Okay, I have Fran with me right here. Fran, slow wave to the audience. How you doing there, my good friend? Fran has been following me, and he's doing a fabulous job of keeping up on my videos, which is a major task. So we're going to talk about all kinds of things. He's following every bit of my research, which is it's extensive, and it's interesting to have someone that is interested in all those things. So anyway, we're going to be going through all kinds of stuff. Today, we're going to start with this. Okay, I got Fran, and he's watching. I am the presenter here, and I'm showing my screen, and he can see this as you can. Now, what we're looking here at that big bar is the Tether experiment. And they notice all these little disk things floating in behind the tether, in front of the tether, and looking like little Pac-Man gobblers. They gobble, gobble, gobble as they go along with this little cut at the bottom here. And sometimes this is top, sometimes this is, it can change, but they go swishing back and forth. And we're going to see this right now. I'm going to show you the video of this, and then I'm going to show you what they just released today. And I haven't shown this to Fran yet. I want to see what his reaction to this is as well. All right, you ready, Fran?
Yeah, I'm ready. Let's go.
Okay, here we go. All right, so Fran's ready for this. Are you ready for this? Here's what we got. They went out with a tether, and a tether just means a big long strap. And it was supposed to run out for more than 10 miles. And when they put it out, it was the size of like an electrical cord. Think of it that way. And that's all it was. They wanted to see if they could gain energy in that. And when they put it out, the signature, as you can see, this big long bar here, they're looking at this. It's a mile wide. That's a mile wide. That's just enormous. Now, you see this particle back here that has this little cut in there and a blank hole in the center? Let's watch that thing move as it goes by that shuttle. And this was by that tether. And this was all taken by the space shuttle that was out there right down the street from this, watching it, out in space. Here goes. So here goes the thing, and watch it. Oh, all right, now, you see I just stopped it? That thing is behind the tether. It's behind the tether. It's not a piece of debris in front. So think of this. That tether is a mile wide. A mile wide. How wide is that thing out there? Look at the size of that thing. It's got to be 3 or 4 miles wide. If this is in front of it, you're right. You follow me, Fran?
Oh yeah, it's got to be at least 4 miles wide, I would think.
It's somewhere in that. You would think so. And they are like gobbling with that little cut there. And sometimes it gets big, sometimes it gets small, and they float by this way. Look, here's one here, really close up. Now, they're watching it. Well, this guy's going to comment about it. Hold on. Okay, so Fran and I are going to be examining this tether experiment and the particles that they saw. Now, this is from a place called Nakun, N-A-K-U-N, and it's 18 years ago this video went up. And nobody has had an explanation for this. And they still don't have an explanation for this. So we are going to see if we can provide that. All right, so what are we looking at? Let's just understand what this tether experiment was all about. They are in the region of space called the Ionosphere. And the Ionosphere is a region in space where nothing exists, basically, other than the white part of photons and electrons. Electrons are dipoles, and they're made of a black and white particle. And this is where all the white particles are. They have no mass. For some reason, they have no mass whatsoever, but they got a hell of a lot of energy. And they are really strongly want to attach to anything that has dark matter within it, which is basically anything that's material that we can touch and it has substance. The only thing that doesn't have any mass to it is where they are right now, is in this ionosphere where there's almost no mass. But there are some particles here that we can see floating around which are massive. They have mass. Now, this particular tether that's running down this whole line all the way down here, all right, that started out at the size of about an electrical cord. Very, very small, no big deal whatsoever. And they expected to see if they could collect any electricity as it passed through space out there, the ionosphere, which ions means electrons. So they're expecting maybe they're going to get a little electricity, and they've got a monitor at the end collecting the electricity, how much is coming through there. Well, all of a sudden, the thing starts glowing like this, a mile wide. Instead of that wire being the size of an electrical cord, it's now 1 mile wide. And they're just going, wow. They didn't know what to make of this. Now, I am going to play this. And you ready, Fran?
Yep.
Okay. He's listening. He's thinking, let's pay attention to what we see. I'm going to stop this on here and on and off, and Fran and I will discuss it, and you can think of what you think. If you got questions, write them down, and we'll try to address them in another video. But here goes. So here we go. I might have to turn it up or down. Let's go. All right, you see all these little particles? You see that one right there? It's behind the tether. There's no question about it, right, Fran? Would you agree?
Oh, that's right behind it. It's not in front.
Yeah, no question is behind it. You can't miss that, as far as I'm concerned. All right, so here we go. We're going to continue. It goes behind, and it's big. Don't forget, the tether is 1 mile wide. Now here's a bunch of other ones coming by. They're all...
Franklin, we see a long line, a couple of star-like things, and a lot of things swimming in the foreground. Can you describe what you're seeing?
You hear that? Can you describe what they're seeing? Now, don't forget. I'm sorry. Go ahead, Fran.
No, they see it.
Oh yeah, they can absolutely see it, and they just dismissed it like, "Whoa, well, it's one of those things." This is what drives me crazy. These are supposed to be scientists. Now, listen to this now. Let me turn this up just a smidge. I got you can see the text, right? Yeah, you can.
Yeah, I see it.
Okay, here we go. I'm going to play this. Here goes.
Well, the long line is a tether, and there's a little bit of debris that kind of flies with us, and it's illuminated by the sun at such low angle. So there's a lot of stray light, and it's getting washed out quickly, but Claude is trying to do a quick, good job here adjusting the cameras.
All right, now, let's address those statements. Number one is there's a lot of these things flying with us. They're not flying with them. Those are behind the tether, which they are observing from, I think it was 80 or 90 nautical miles away in their space capsule watching this event. So the particles are behind. They're on the other side, so there's nothing floating with them. Do you agree, Fran?
Oh, yeah. Yeah.
So you just can't miss that if you're thinking. So there's not something going with them. And they see them blinking on and off, and they all, not all of them, but a lot of them have this round dish shape with a dark hole in the center and a little cut on them, which it seems to almost like gobble. Now watch, let's just keep an eye on this. Here it goes again. So again, I'll be stopping it here and there. And don't forget, that tether is 1 mile wide now instead of the size of a electrical cord. And the reason it's so wide is because all the white particles want to get to the black core, which is where there is mass. And it's just floating. That normally doesn't happen out there. Normally that stuff just gets burnt up, and it will. And that's what happened. It was a failure of the material. They say, oh, well, it was a flaw. No, it was no flaw. It was just overloaded with electrons. So here goes.
Copy that.
Again, that description by the crew, this is the
Behind,
tether,
again.
the satellite with approximately 12 miles of tether still attached to it.
12 miles of tether, all right, of that particular wire. Now listen to this.
Columbia and the satellite now just passing over the west coast of northern Africa. The two spacecraft now 90 nautical miles apart.
All right, you hear that? 90 miles apart, they're looking, and these things are going behind that
The
tether.
satellite did have communications with it during the close pass between Columbia and the satellite.
You see that tether is just humming with particles.
Columbia, Houston, that's a much better view, a lot more contrast visible. And how wide?
All right, I want you to look at something. You see that little round one that I'm circling there, which has a bright spot and a dark spot. Another one here, same thing, bright spot, dark spot. They have a little lump hanging off them. All right, when they see them from this distance, they can see the bright spot versus the dark spot. The dark spot is going to be much bigger than the bright spot. Now, the tether, as you can see, has absorbed just as much of particles as it can possibly hold, and that's when it exploded. So here it goes.
Wide does that tether appear to be? It seems to resemble a much wider strand than we'd expect. Can you describe which way the
A much wider strand than we would expect. That's because it's just all they're seeing now is the electrons bouncing back at them, glowing. All right,
satellite is visible on that strand?
He's not explaining it because he can't explain it. How could it be a mile wide when they started out with something the size of an electrical cord? They just can't explain it, so they don't respond.
It was like a piece of spaghetti growing to be a mile wide, and then it snaps.
Exactly. It's just, and there's nothing they can say about it. They have no. Now, you see all these particles? They're all very, very similar, those little round particles, and they're glowing and ripping.
Satellite now.
They're all like the same size,
100
too.
nautical miles.
They completely unzoomed, and you see the full extent of the tether. I tried to adjust the focus, but I can't get better than that.
Okay, Claude. Thank you.
You're going to zoom in now.
Now they're going to try to zoom in. Let's see what they see. They just give up. All right. All right, this is another shot. This is Associated Press, apparently. AP Archive. 5.79 million subscribers. Now, this is when they're doing the tether experiment and the tether breaks. And this shows them unwrapping it and as it breaks, I think. We're just watching it for the first time. I just clicked on it, so we're going to find out what it shows. Here goes. Let me open the screen up, and like I said, AP Archive. And here we go.
Meters of tether left in the boom.
Okay, this is taken from the EOS 350.
Whoop, hold on. Hold on, Fran. I got to turn this up. It's gonna. Here it goes.
The shape of the tether, and you can see it just starting to go slack now. Unfortunately, in all the excitement, somebody bumped the camera, and we lost it. Okay, and that's...
Look at that. That's the tether. Now, don't say anything, Fran, because it's so loud right now that you'll blow my eardrums out. But that's the tether. We'll talk about it in a second. In the meantime, hold on one second. Okay, Fran and I are back again, and they are unwrapping the tether right now. And that is the wire. It's supposed to go, I think, 12 miles long or something like that. And you can see it's starting to glow like crazy because it is all the electrons that are out in space, and there's nothing but electrons where they are. There's nothing but these glowy particles. And they are attacking that dark matter because that's their mission in life. It's just like lightning wants to get to ground. This is a floating ground. It's floating in that area, and they see it as ground. So they attack it with just gobble all over it. Now, let's watch it. So they're unwrapping this tether.
We had of it as it was drifting away, and we got it for a minute or so before sunset.
From the vicinity of the shuttle, the satellite is continuing to move away rapidly from Columbia.
Bravo, we had the camera Bravo constantly on the tether. Unfortunately, we don't see the tip of the boom, but in a few seconds you'll see the tether break. And this one, of course, was not attended, it just recorded. It could not be bumped either, so it will record steadily the whole break and
You see up here at the top, I believe at the very top is where they have the capsule that let this tether out, and it has a like a voltmeter up in there to see how much current and how much voltage they're coming. And they ended up with 3,500 volts, I think. And then it just basically the wire exploded because it only could allow, I think it was only getting a half an amp. But that's because of the restriction of the wire, not because if you got 3,500 volts, you're going to get a hell of a lot more amperage than that. But that's why the wire failed, not because they had some failure in the insulation. That's what they said. Oh, there must have been failure in insulation. No, I don't agree with that. It's just too much current. So here's what happens. They're recording this steadily while the whole thing and the coil unwinds, and then it's going to break.
back to the tether.
Copy, Claude.
Now that thing ends up being a mile wide when it's, because look, see it just snapped. I believe it just snapped. You see how it's glowing like crazy?
Well, hold there on the bottom of the boom for a minute there. We're trying to show you there's a little bit of a little bit of tether at the bottom of the boom. It's real difficult to see, but it comes out, bends back down, and then comes back up again. So we're not exactly certain.
All right, what they're looking at now is they're actually focusing in on the actual tether. That's what they're saying. And there's just a little piece, they said, coming back. Let's back it up here. All right. Let's back it up to where, here it says, on the bottom of the moon for a minute. On the bottom of the moon. I don't think that's an accurate, hold on. It obviously broke right in that area there somewhere. And
Well, hold there on the bottom of the boom for a minute there. We're trying to show you there's a little bit of
you know what they said, the bottom of the boom, not the bottom of the moon. That's what happened there. They're looking at this, and it's the bottom of the boom that was letting this out.
a little bit of tether at the bottom of the boom.
You see, at the bottom, a little bit of tether at the bottom of the boom.
It's real difficult
See,
to
boom.
see, but it comes out, bends back down, and then comes back up again. So we're not exactly certain how much is laying there. We're setting that up there. We'll try to zoom in here with the camera alpha.
Yep.
Oh boy. Hold on. All right, so we saw that little particle there. You can still see this, right, Fran?
Oh, yeah.
All right, so you see the particle with the little cut in there. That's the particle that I just saw something today that looks like it might have some similarity to this. Now, The other ones we saw floating around in space not only had a cut in them, some of them had a bubble sticking off the side. Remember seeing those, Fran? Where they had they were bouncing around in a circle with the bubble on it?
Well, is that where you said it was that's the black part? Because it's...
Yeah, there was like, they were like little dipoles floating around in circles. Hold on, let me just be sure everybody sees that before we go any further. Okay, I want you to understand there is I'll show it to you and try to explain it as we see it. But here is these big ones, the big round ones with a spot in the center, and they have like an open mouth to them. In addition to that, they also have ones with bubbles on them. Now watch this.
Controllers for the satellite did have communications with it during the close pass between Columbia and the satellite.
All right, now what I want you to watch is these glowy ones. And they're all over, and they're spinning, and you can see a little, like you can see that they're dipoles. Watch down at the bottom ones, down where they're flipping and they're turning white. You see them over here?
Columbia, Houston, that's a much better view, a lot more contrast.
You see down here?
And how wide does that tether appear to be? It seems to resemble a much wider strand than we'd expect. Can you describe which way the satellite is visible on that?
All right, they are saying, "Wow, look how big that sucker is." Yes, but also you can see these right next to the tether. You can see a black and a white one. It's not black and white. It's really white, and the other part of it is not white. It's black, but you can see it. It has a glowy edge around it, they call it, because it's being illuminated. You see them how they're blinking? You can see that, right, Fran?
Oh yeah, no, they're turning off and on.
You see them all of them blinking on and off, basically.
They're all the same size.
There's big ones and small ones, but they're I'm not sure. Maybe they are all the same size. That's a possibility. Hold on.
I mean, you don't see ones larger than that.
Well, you could very well be right, because we can't really judge. We don't know how far one is in relation to the other. You follow me?
I'm not up there.
Yeah, we know that one is going to be way behind the other. Sometimes they come in front of the tether, sometimes behind. They could all be the same size. That's a good possibility. And the brighter ones are just very much. Yeah, well, let's take a look at something that I got in today that might relate to this. If the universe is completely living, and this is nothing more than almost like cytoplasm in a creature's body. Hold on a second. All right, my friends, you see what I'm pointing out here? Cytoplasm. Well, what is that? It's the material within a eukaryotic or prokaryotic cell. It's the fluids within a cell. It's really everything, but it's primarily the fluids. It's enclosed by the cell membrane, including the organelles, which if we're living inside of some gigantic creature, we are literally viruses and bacteria on organelles. Let's think of it that way, just for the kicks. It's just as a thought experiment. All right, so let's just say we're living inside of a creature's body, just like things are living inside our body. We know that for a fact. There's bacteria, thousands and thousands of different bacteria in us, viruses, funguses, all kinds of things, doing chemistry inside our body, just as we are doing chemistry on the face of this planet. Let's just think about it that way for 1 minute. Okay, my friends. As you know, or may not know, in 2015, working with my mud fossils, I found the layer that they now call the interstitium, which is a fluid-filled highway. Now, this is today, just came out previously unknown loading dock found inside human cells, which we never knew about. Well, it came out June 26, 2025. Now, here's these little organelles. And here's that little bump on them. You see what I'm pointing at, Fran?
Oh, yeah.
All right. Now, these are the organelles, and there's a bump. Now, they found this scientist at the University of Virginia, super small, specialized structure, has a role recycling materials inside our cells. The discovery could lead to improved treatments, wide range of diseases, just as our bodies have organs that carry out specific functions. The organs themselves are the housing primarily, and they have the equipment inside those specific organs and cells. But the bacteria really do the job. The bacteria create the enzymes. So they're just basically living in these little factories. Now, and that's what keeps us healthy and alive, is literally the enzymes. I've shown a lot of videos on that. Now, each of our cells has tiny structures, which is the little factories, which are called organelles. Some of the more well-known organelles include the mitochondria. That's where your energy is produced. That's what we've been talking about a little bit, Fran. And that produces the chemical energy for your biology to run. Then there's the ribosomes, which I talk all the time about, which contain the enzymes. And they put proteins together. The enzymes are the builders of the proteins. You don't have the enzymes, you don't get the proteins built. And if you don't have the enzymes, it means you don't have the bacteria. The bacteria start everything. All right? So the mitochondria produce energy, and the energy ribosomes put proteins together, and the nuclei, which store our genetic material. So this goes on and on and on about this stuff. But what I want you to see is this new factory they're talking about, which is this new organelle, that round thing with the little blob on it. Now, let me show you some more I discovered about this, but this is like today's news. Hold on a second, because this is a brand new organ they never knew existed, and it is a recycler. Primarily like your lymph nodes, recycle debris that's in the lymph fluids. They are the recyclers. These recycle inside the cells. All right, the lymph nodes recycle outside the cells. These are inside the cell recyclers. That's the way I'm taking it so far. Like I say, it's pretty brand new stuff. Hold on one second. You ready, Fran? You got any questions about this so far?
No, I'm just thinking that notch, the pizza slice, looks just like that.
Yeah, and there's the notch. Where did I show you some other shots I saw? As a matter of fact, here, hold on one second. I got this thing. I'll bring it up right now. Okay, I got Fran again, and we're trying to make a determination if this makes any sense. We saw empty space, more or less empty, and we saw these little things floating around. It looked very similar to these. There's one, some of them have bumps on them, like we saw spinning, and some of them have little mouths on them, like this one over here, that we saw, like, looked like a little Pac-Man going through the, eating whatever they eat. I don't know if they eat, they must be eating something. And they're glowing. All right, in space, we know they're glowing, so there's no question whatsoever, in my opinion, that light is bouncing off of them. Light only bounces off of matter. If there's no matter, there is no light. And mostly out there is blank space in that section, but there's enough of these particles floating around, and we can see them, and they're material particles, just like that tether is a material substance that has matter to it. And that's why all those electrons are flocking into that material substance, which was the tether, so much that it exploded the tether. Because there were so many electrons, it couldn't handle it. Now, what are these things doing? What are these little jobbers doing? And they're big, they're not little. Right, Fran, what did we decide about 4 miles?
They all look about 4 or 5 miles.
Something like that.
They all look about the same size, just like this right here. And then also you have the small ones too.
Right, well, these little brown ones appear to be what these big green ones are eating or doing something with them. I don't know what they're doing, but these are what's called hemifusomes. Hemi is blood. These are in the cells. These are recyclers, they call it, of whatever material is left over, and it appears to be these little red balls of iron are left over after the nucleophilic substitution and invasion, probably of the oxygen. Something's going on with these little, and I haven't really looked into it, and nobody has yet. This is brand new. Nobody even understands it yet. But they do see these things, and they're calling it now the hemifusomes, which is a new organ that all right, here's what it says right here. The green and orange structures in this image are hemifusomes. That's these big round green ones, and they look to be exactly the same size. As you mentioned before, Fran said, they all look to be the same size, and at first I said, nah, you can see there's big ones and little ones. However, if you think about it logically, they're different distances, so they're going to look bigger and smaller. They could all be exactly the same size, right? They could be. So let's say they're hemifusomes, which is what they're calling them now. Newly discovered organelles may represent a previously unrecognized pathway for recycling inside of human cells. These are inside human cells. They never knew they were here before. And they are the recyclers of whatever these brown things are. That's the way I'm seeing this. What do you think, Fran? What do you think? That makes sense?
They're eating something.
That's what they're claiming. Yeah, right now there's just not enough information about them, I don't think, to make a determination what's going on. But I see the same look to them as we're seeing in space, which would be considered to be a cytoplasm if the universe was a living universe. And we, as planets and solar systems, are little individual energy, cells. We could be literally a cell, and the mitochondria could be the sun giving off the energy to run the cell. And these little things are like light particles, because they never even knew they were here, so they got to be so small that, some of this stuff, you can have millions and millions of these particles inside of one single cell. Ribosomes is a perfect example. Ribosomes have the enzymes, and they are so small that in those ribosomes you could have millions of them, millions, inside of a single cell. And not only that, you still have all the other stuff that the cell uses to do its function, the organelles, the mitochondria, the this, the that. We could literally be inside of a single living cell. What do you think, Fran? Is that you?
It's just scaling up. Makes sense.
Well, nobody knows anything at this moment. I can tell you that right now. Everybody's clueless, and including me. I'm learning, but I'm seeing things that, they're scary, but you gotta look.
Everything's biology. Everything's biology.
It seems to be. These hearts in space, Bennu, I've shown it over and over, it's heart muscle. And they just can't deal with it. That's the scary part. that the people that teach us what we have to believe can't deal with reality. That's the real scary part. Now, I don't know if this is real or not, what we're thinking, or what I'm thinking.
It's real. I'm thinking the same thing.
It sure has the look. You know, you got to start by seeing the look. And this is, it appears, an actual picture. You know these little, excuse me, these little square lines here? That must be pixels or something inside of this picture. As far as I can tell. Hold on one second. Okay, so this just was discovered, these hemifusomes. The biologists discovered previously unknown organelle complexes inside human cells. Now, we were just looking at this, trying to determine whatever we can determine about it, because this is all brand new stuff. Each one of these different colors is a different type of organelle. And we see these little balls here. This isn't, the other thing is this is cryogenic. That means it's frozen as close to absolute zero as you can get. And these balls appear to be at certain equal distances. And I think we understand why, because they basically crystallize when they lose their electrons. You know, they form a grid pattern. Let's just go with that. Now, they're saying they've discovered these new little tiny organelles, and this what they call the hemifusome. Heme is blood. This is inside cells. These are inside of cells, and there's a ton of them inside of cells. It appears to me, that's what I'm seeing. Now, it says this is like discovering a new recycling center inside the cell. They always thought it was just lymphatic and lymph nodes that did the recycling in the body. And of course your liver and your so forth, kidneys clean up a lot of stuff. But this is a new recycling center inside the cells, not outside. They always, I don't ever remember hearing anything about, well, that's a good point that I'm starting to think about, is the ribosomes are inside the cell, and you can have 1,000 ribosomes, 1,000,000 or more, inside of one single human cell. So they're doing the same job as these hemifusomes. So maybe the hemifusomes actually are the ribosomes. I got to think about this. They're the recycling center inside the cells. Are they the enzymes? We got to look into this. And they're talking about rare genetic disorders. Genes are turned on by enzymes. Enzymes are created by bacteria. Now, problems with how cells handle cargo are at the root of many disorders. Yes, that's what I've been talking about. You don't have the bacteria, you don't have the enzymes. You don't have the enzymes, you can't handle the cargo, either in or out. Right? We've been talking about this a lot, right?
You stuff to do the quick chemistry.
Yeah, I've been showing over and over how the enzymes are handled this stuff. That's what they're looking at. This new organelle, I don't think it's an organelle. I think it's the enzymes doing their job. Now, are they little organs that are facilitating the use of these? Like, see, they believe these organelles, which are these little hemistomes or whatever they call them, facilitate the formation of vesicles, which are tiny blister-like sacs, and they act as mixing bowls where the organelles make up multiple vesicles. So this is where they're shooting out their contents to attack things. You can think of vesicles like little delivery trucks inside the cell. Well, maybe. All right, they're saying the hemifusome is like a loading dock where they connect and transfer cargo. That's what the enzymes would do. They're just looking at enzymes, I think. Such insights could lead to targeted treatments for a range of serious genetic disorders. Yes, if you don't have the enzymes, you're going to have the disorder. This is just the beginning, Dr. Ibrahim said. Now that we know hemifusomes exist, we can start asking how they behave in healthy cells and what happens when things go wrong. Well, it means you don't have the enzymes, so the cells cannot create the products they're supposed to be creating. Or if you don't have enough of the bacteria to create enough of the enzymes, you don't get enough of the response. So you have an extremely slow recovery. Or you may not recover at all because you don't have enough bacteria to fight off the invaders or the thing. And sometimes you just don't turn on the genes that are good, that are inside of you. They're working fine, but you just don't have them turned on. There's a lot we can learn from this. And this is interesting. I didn't realize this when I started. What do you think, Fran?
This is amazing. No, I get what you're saying, and
I think
looks like you're just looking at it, you know what I mean?
There's so much to this. This is inside the human body, how you heal and how the enzymes affect everything. And we're seeing almost the same looking things in space. This is just too much for my mind. And once you factor in the fact of asteroid Bennu being a heart in space, and Psyche is also a heart in space, and that, no question whatsoever. And they have a piece of that heart, and its sarcomeres, and its membranes, and its phosphates, and its DNA and RNA, and 16,000 organic chemicals, sitting right here on a desk somewhere, all over. They got enough of it, they sent some around, and they showed the samples. They're heart sarcomeres. And the chemistry and the DNA and the RNA, it's all there. And they cannot accept what they see. This is the key to why we've had such a disaster in science and moving forward. They just don't understand any of this stuff, and they don't want to understand it. Very discouraging. But does this make sense?
Knowledge of what a rock is.
Well, the problem is
it comes down to what is a rock, and if it's a body part, that changes everything.
Yeah. Everything that was told in the ancient texts and so forth, and especially like the Bible, which I don't believe everything that's in the Bible. Absolutely not. I believe the Bible is a cleaned up text that talks about mythology, which is not fake. It's not something that didn't happen. Mythology is just ancient history. And the Bible drew on that, but the original source was much earlier than the Bible. And, Enoch and all the things they discarded because it was just too spectacular for the average human mind to even consider. And as I present these things today, with the evidence we have now, and the things I have shown, they're undeniable. And my stuff is giants, and the DNA is done, and the CAT scans, and nobody's questioning it. They just won't speak about it because it destroys their authority to tell you what you have to believe.
And everything that they're selling you, and you got to start, then we got to come here and start over because everything they told you was wrong.
It's at least in question. Now, a scientist questions things, and they answer questions, and they seek answers to questions, This is not what academia does. Academia dictates what you must respond with, word for word almost. And if you can respond with exactly what you were told, then you're a genius. But if you can't respond with exactly what you're told, you're degraded for every question that you create about what you're being told. Every time you ask a question, trust me, you don't get more points, you get less points. Because you putting mainstream in a corner they can't get out of. They can't get out of this corner now. There's no way out. And the only thing they can do is to disparage those that are asking the questions that they should be asking themselves.
You interacted with them. Like, what did they... I know you interacted with them, and you went to school. Like, you went undercover, and you broke into the schools and learned everything that they were teaching to ask them questions about the stuff that you know, and they still dismiss you, and they call you a whack job when that they want to teach you, and you know it's wrong.
Well, there's literally no question now that they know what I am saying is true. Even Yale did a paper a year after I presented all my stuff to them, CAT scans, DNA, everything, to Derek Briggs. They did a paper saying that there was exceptional preservation of soft body creatures, and exactly what I said, because of silica-rich oceans and the silicates invaded and so forth. They turned it around. Instead of mud, they said sand, which is just not true. Sand is the product of the invasion. Sand is literally blood cells in most cases that have been attached to silicates. The silicates is the key. And it's because of the impact of Venus with Earth, bringing up all that siliceous ooze. Just look it up. It coats the ocean basement layers, and it came up during the flood and literally created all the gemstones. And they attached silicates to blood and so forth. That gives them all the colors, transition metals in the blood. And all of those silicates created the mud fossils. And Yale made a paper a year after my paper saying exactly what I said, basically. Exceptional preservation of soft-bodied creatures promoted by silica-rich oceans. As a matter of fact, I got it right here. You won't be able to see this, but everybody else, well, you've seen it enough times anyway. Here it is right here. This is the Yale paper, 2016. I was trying to work with Derek Briggs, this guy right here, Derek Briggs, one of the authors, for years before this, and I had sent him all my stuff, and he wouldn't do it, so I went and had it DNA tested and CAT scan and everything, and they still dismissed it. And then I presented all this stuff to NASA, too, because they have the same stuff on Mars as they have here on Earth, exact same stuff. And NASA ended up funding this stuff from Yale. NASA ended up being the funder of all this stuff. And I had, like I said, I had presented all this stuff to NASA. And I was surprised to see that they were the ones that funded this. And all these people I'm showing, you're not seeing this, I know, Fran, but all these people I'm showing, I sent my information to most of them. And they're showing these samples of this and that, and how they found all these little things, and basically everything I sent to them. And NASA ended up paying for it, so. All right, now, here we go. So, that's it. I got Fran up here. What do you want to do next year? We're talking about just one thing after another, man. It just never stops.
Boy, oh boy.
Hold on one second. Let me think. Okay, as we go forward, things just change. You know, they change. You have to start thinking, what does this mean in a total perspective of things? This Hemifusome, that's just brand spanking new, right off the press. Now, we're seeing that particular looking thing inside of a cell. Now, if that is inside the cytoplasm of a cell, let's go and see what else is in a cell. Because, like I say, nobody's got any answer for how big the universe is, how big the solar, we can guesstimate this and that, but all this stuff about the Webb telescope telling us how far things are, that doesn't work. They have now agreed that space is saturated with particles. Light does not, and they say light does slow down. This is just brand new. You've heard of this, right?
Oh, yeah.
Yeah. This is brand new, too. And that was a study, I believe it was Harvard that did that. It might have been MIT, but I think they all agree that they can see light shifting from these rapid bursts, radio frequency bursts. And they can see light and literally slowing down in space. So it just doesn't happen just good. That one, light slows down. Light is a frequency, and a frequency bumps and bumps and bumps and just keeps slowing down. So basically that's that. Now let's hold on one second. Okay, Fran and I have been discussing what this has any consequence if the universe was alive, and nobody, there's not a person on the face of the planet can say what the universe is constructed of. It's just there. Now, is it a living creature? Which I haven't found a single molecule that I can say didn't come from life, not a single one. And light makes up all matter. It's called atomic vapor, and it comes from light. Atomic vapor, and it condenses into matter, just like water condenses into, fog and so forth condenses into water. So we have seen the things that are out there in space, these little round jobbers gobbling around, and we see the same thing, this brand new thing just came out a couple days ago, or very recently, these hemifusomes. Can we draw any relationship to that? I think we can. I'm going to dig right into it now. I'm going to go into the presenter screen mode where you're just going to see my screen. So let's start by looking at what a cell is. Because we could literally be inside of a cell, and the sun could literally be the source of energy, which would be the mitochondria. It sounds insanely crazy, but let's just see what's inside of a cell. Okay, this is an animal cell. Now, which we are animals, so we have this type of a cell structure. Now, these little holes are in the membrane of the cell, which allow things to come in and out. The cell membrane allows entrance through these pores to come in and out of here, and there's some kind of enzyme that controls who can get in and out. There's some kind of intelligence. It's not just anything can float in and out. Now, that's number one. That's how you get inside this system. Let's call it a system. And if it was a solar system, well, I guess the nucleus would be, the sun, and all these little organelles, and centrosomes, and lysosomes, or maybe even the mitochondrion, and the secretory vesicles, and the perioxomes, and all of these other things are things that are inside of a cell, which we're seeing all kinds of things inside of what we would call our solar system. And there's all kinds of little things floating around out there. I mean, just gobs and gobs of things. And as I spoke before, there's things called ribosomes. All right, now we gotta see this little thing right here. I'm circling up at the top, ribosome. They don't even show them because they're so small, you never see them. They're inside of this one single cell, and we have trillions and trillions upon trillions of them in our body. Inside this cell, There's millions of even just the ribosomes. Millions. And every one of those is a chemistry set. And when I say it's a chemistry set, it's an enzyme. Every ribosome is created by a bacteria and ends up being an enzyme. And that enzyme has chemistry in it that is beyond spectacular. So think of how complicated this life device is here. And then, of course, you got the rough endoplasmic reticulum. This right here is where these little balls, the ribosomes, collect. And it looks like they collect around the entrance into the nucleus. So these ribosomes are enzymes that are gatekeepers. It appears to me, because you see all this secondary ball, the outside ball has its own membrane. That's the membrane to inside the cell. And then this is another membrane outside the sun. I mean outside the outside the central core, which happens to be the nucleus. Now, if it was in our solar system, that would be the sun. And then all these other things are just things that are floating around in space.
Would that core be dark matter?
Well, yes. Yes, it could. Yes. Now that you mention it, given a second to think about it. Well, let's think about this. Yeah, that's a good point.
Is it throwing?
Well, let's
create electricity or something?
Well, we know that dark matter attracts the white matter. We know that. And I have shown you have you seen the pictures that the Russians did in space where they have a black core and all the little white particles are surrounding it?
It's a black hole.
Did you see that?
Yeah.
Okay, so you have seen that. And it mimics this right here, where my mouse is circling, would be the black hole, and the white particles are surrounding it, trying to get to that black core. Now, it's a similar situation. What it does inside this nucleus, I don't know. But it could be the source of energy. Now, they always talk about the mitochondria being the energy source because it creates something called ATP. And that's a phosphate. Something tryptophosphate, I believe. I got to look back into this. So as we talk, I start to remember things, and as things start to make sense or make less sense. Now, I see, yeah, that could very well be. This could be the corona. That could be the corona of the sun. Because the sun is 10,000 degrees and the corona is millions of degrees because it's feeding off energy to everything that surrounds it, whether it's us or all the other planets or everything that's out there. It's spitting out particles. You follow where my mouse is going?
That's it.
All right, it's feeding this as a source of energy, because every bit of energy, there's not a single bit of energy that is not made of dipoles. The dipole particles are the energy source of everything. Everything is run by dipole particles. And it's the breakdown of the dipoles that gives off the dipole electrons. Those are the actual energetic particles. You have to get down to that level of atomic vapor to add or subtract energy, or to make or break electrical bonds, which make or break particles, which are atoms. They break them and make them. And that's what's called isotopes. And that's decay. And decay can go both ways. Decay goes downwards and it goes upwards. If you have enough heat and pressure to something, it will decay upwards. It won't decay downwards. if you have cold and a lot of fluids running through it, and there's no extra energy being added to it, they will decay downwards. That's the nature of the stability of every atom. Did that make sense?
Oh yeah.
All right, so atoms are stable at a certain number of dipole electrons. All right, and the first stable number is hydrogen, which is 1840 approximately dipoles. Now, the size of the particles that we're talking about is just staggers the mind, absolutely staggers the mind. So a single cell, all of that stuff in that cell working, and then millions and millions of these ribosomes, which are the chemistry sets, which have enzymes in them that are so fancy you couldn't possibly imagine. Let me show you something. Don't forget what these cells look like. And don't forget what ribosomes are. These little tiny particles, you can't even see them. And they're free floaters. They don't eat. They don't poop. They're just like pills. They're just like pills floating around in your body waiting to get a tap, which is a trigger. The trigger hits it and says, hey, I need you. Come over here. And it comes over and blip, pops open and does spectacular chemistry. Let me show you that. Okay, once again, Fran and I have been talking, and as you know, I said we got ribosomes. And when you have ribosomes, you got enzymes. But the only way you have a ribosome is to have a bacteria that creates it. Now, you got anything you want to add to this at this point, Fran?
Let's go see what's going on.
All right, so let's just start right into it. Excusez-moi. Now, here is the deal. We have, I don't know how many thousands and thousands and thousands of species of bacteria living in us, primarily in our digestive system, because they do the chemistry, and they do the chemistry using ribosomes. Now, you see that little white block right down here? That little white block is a ribosome, is where ribosomes come from. Just think of it that way. This cell is a single cell. Bacteria are one cell. One cell. Their DNA controls what they look like, and if they have a flagella, and it has one little lump of DNA that says, here's what I produce. This is my job in life, is to produce this ribosome. And a ribosome is this chain over here, you see it, the chain of polypeptide amino acids. All right, these are the amino acids, all these little different colored balls. And I think there's like 20 or so amino acids. I think maybe 28, maybe, I can't remember now. But there's a lot of them. And every one of them is another program code. Now, you work with programs, right, Fran?
Mm-hmm.
Now, I don't know what they use now for bits and bytes and so forth, but when I was in this, there was binary code, there's ones and zeros. Each one of these little bits can be 20 different types of characters, and each character is so complex, it's amazing. Each one of these is an amino acid. So that gives this program unbelievable capability. Does that make sense, Fran?
It's a lot of code.
A lot of code. Because a lot of code. Because every ball can be 20 or more characters, whereas it used to be ones and zeros. You had two characters on and off. Here you can have all kinds of different states attached to all kinds of other states, attached to all kinds of other states, and if they don't all follow this exact same pattern, it's what they call denatured, which means it's not going to work. So it comes up, and all these balls are these polypeptide chains created by the bacteria due to its program code that attaches to its body code, and says, I make this particular ribosome. So it comes out, and here, these little things going up and down are just like keystrokes, just like a keystroke into a typewriter ribbon. And it plugs in. Anywhere you see white here, that's an enzyme. And this is called transferase. Anything you hear, every time you hear A's on something, it's an enzyme, ASE. And this is transferase. And these different types of amino acids, and they come down and they program this messenger RNA, mRNA. It's a messenger. It carries the code. Doesn't do anything until it gets...
Is it attached by charge?
Say that again?
You know, it's attached and everything. How are they hooking up? They're hooking up by charge?
Yeah, the same way DNA works. DNA works where the C's and the T's or the A's and the G's or whatever, CTAGs. You see the AGTGT up above here?
Yeah.
All right. They have to be in a certain order, and then whatever order these are in will force the other side to attract to it. So that's why you get the double helix. But when you're going to reprogram your DNA, you see the DNA coming in over here?
Mm-hmm.
And it's double-stranded. That's your normal DNA. Everything's good. You're walking down the street and you get bit by a tsetse fly. And your body responds by saying, whoa, we got a problem. And we got to create some chemistry to deal with this problem. And I don't know how fast it works, but it works pretty damn fast. And the bacteria get reprogrammed by a virus. All right? The virus, as they say, takes over the program of the DNA of the bacteria cell DNA, which is, so what happens is a virus comes in, and the bacteria somehow, either the virus comes in, a good virus, and says, hey, I just found a bad virus guy that just came in, and I'm going to reprogram you to go out and attack that sucker, or he's going to kill us. So the virus comes into this little block, and it says, I'm the new guy here, forget the code you used to work with, and he sets up a new code. And the DNA starts squirting out new ribosomes, which go to mRNA and the messenger RNA, and they float around and they cut into the regular double helix RNA, a DNA, and they replace a section. All right? Now the U, you see the U here? The U is uracil. And that's like the CRISPR. I'm not sure exactly, I don't want to speak over my head on this, but just take it this way. If you got uracil, you can cut into the DNA and replace a little chunk of the DNA with, see, polymerase? Those are the polymer acids, you know, polymers. And it erases them and replaces them. And that happens inside this enzyme. The cloud is an enzyme.
So U is a knife?
Yeah, yeah, that's a good way to put it. It's a knife.
U cuts in, and that's what starts the reprogramming. Right,
uracil. Yeah, uracil is a key component of reprogramming DNA. Hold on one second. I want to be sure I'm telling you the right story here. Okay, I'm back with Fran now, and I mentioned about uracil, and they claim it's not, it doesn't snip into the DNA, but it's how you look at it. In transcription, which is what we're looking at, was transcription. During transcription, where DNA is copied into RNA. Well, that's not correct. It's RNA is copied into DNA, actually. Uracil is incorporated into the mRNA strand, which is what I said, complementary to adenine in a DNA template. So it basically somehow is working to get you into the DNA. To me, it works, it seems to be. And then they say in DNA repair, uracil can be mistakenly incorporated into DNA during replication. Uracil, but the enzyme UDG removes these erroneous uracil bases from DNA. An enzyme. It can fix DNA. That's an enzyme. It can fix DNA. They always talk about, oh, your problem with your genetic code, your DNA. Well, this is uracil DNA glycolase. An enzyme, UDG, removes the erroneous uracil bases from DNA. It goes and sniffs out and says, hey, you got some extra uracil in here. You got to get this out of here. You're going to have a problem. But that's done with an enzyme. It initiating a base excision, which is cutting in, repair pathway, which helps maintain genome integrity. That's so your genes are not malfunctioning. But that's done by an enzyme. What creates an enzyme? An enzyme is literally only created by bacteria in your body. That's what creates an enzyme. They just don't spontaneously combust in your body. All right, I got Fran again. We're talking, and I mentioned that bacteria are the factories that create the enzymes. They're the factories. Those little buggers are programmed to create these ribosomes, which are the enzymes. Now, the enzymes are crucial for cellular processes, including the breakdown of complex molecules for food, metabolic, which means life activities. Bacteria secrete enzymes into their environment to facilitate these processes. How fast do enzymes work? How fast? An enzyme is a catalyst. A catalyst means it speeds up. You see where it said it? It speeds up these processes, wherever it said it there. Now, how fast does it speed them up? Enzymes are, can you see this, Fran? Hold on. Now you can.
Bingo.
All right. Enzymes, right here, are powerful biological catalysts. Biological, made by life, made by bacteria. Catalysts. They make things happen very fast. So the enzymes are very powerful biological catalysts that significantly accelerate chemical reactions. Significantly. Now, what does that mean? They can speed up reactions by factors ranging from 1,000,000 to 1,000,000,000,000 times faster than the same reaction without the enzyme. That reaction would occur, but it would be such a slow make and break of processes of chemistry to get to the end result. It would take, they claim, some of them up to billions of years to do the same chemistry that one particular enzyme can do in less than 1 second. Let that sink in. Millions to billions of years to do the same chemistry as is done in 1 second with the correct enzyme. It's a dramatic increase in reaction rate. It's crucial for life processes to occur at a speed that can sustain life. If you don't have the enzymes, you're done. You're absolutely, totally done. You fall over dead, case closed. Have a nice day. Now, moving at the speed of life. It's really moving at the speed of light. It's called click chemistry.
It sounds like quantum entanglement. It moves that fast. It's a trillion times a second. I mean, that's.
Oh yeah. It's just It's exactly like a electric discharge on the surface level. Now, you've seen lightning. Everybody's seen lightning. It comes down from the sky. You can see it actually propagating, moving down towards the Earth. You can see it in motion, right? Would you agree with that?
Seeking ground, yep.
Okay. Now, what if you had a plate that it could discharge to, like, from its complete plate down to another complete plate? It would be trillions of times faster as an enzyme going face to face with other particles. This is going to be hard for you to understand, but if, in other words, you get more reaction when you go surface to surface than you do when you go deep into a surface. You follow what I'm getting at?
When they're touching, you move quicker.
You get more energetic bounce back because it's called the Kashmir effect. The closer you get and the flatter the distance of the plates are between the pushing against each other creates more and more energy because, first of all, it really doesn't matter. This is just silly to talk about it, but it's the enzyme has to make and break chemical reactions in these half-lives trillions of times in a second to move up the atomic chain, which is decaying upward, or move down the atomic chain, which is decaying downward. Depends on what it's working on. But you have to have the enzymes. They have to be floating around. And every single cell has millions of them. That's the key. Hold on one second. All right, I just told you what a ribosome does. It creates an enzyme, which is a gigantic, elegant molecule. Now, in a typical mammalian cell, a mammal, which we are, a typical cell can contain millions of ribosomes. Each one of them is a chemistry set, and ranging from 1 million to 10 million in a single mammalian cell. Specifically, a rapidly growing mammalian cell can have up to 10 million ribosomes. Why does this rapidly growing need 10 million? It's doing a lot of chemistry. In contrast, a single E. coli cell contains around 20,000 ribosomes. Well, we're eukaryotes. A prokaryote is single cell, basically. They're bacteria. So they don't have as many ribosomes. We have up to 1 to 10 million. And when you're really doing a lot of chemistry in your body, you have a lot more. And they are essential for protein synthesis. And that's what we live on, is proteins. That's what builds everything in our body, is proteins. And so anyway, we know we've got millions and millions of ribosomes in each cell. In each cell. Okay. Fran knows this. He understands this. So I'm not speaking to Fran. I'm speaking to everybody else. These balls are the ribosomes. Remember I said they are tiny, 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 tiny little balls. You can have millions of them, 10 million in a single cell. But they don't eat, they don't poop, they just float around. Something triggers them. You see this word trigger? That's something. Tickles them. A piece of enzyme bites into them and they split open and the sheath comes off, which is the wrapping. Just like you take it out of a plastic bag and it just is like a fluff. And it fluffs everywhere. And it creates all these little spiral, squiggly little patterns. Now, sometimes it could be an enzyme that's inside that ball. Now, every ball is created by a different species of bacteria. So it's Lactobacillus acidophilus, Lactobacillus reuteri, Lactobacillus, a ton of them, probably thousands of them, that start with the lacto, which is milk, works with milkish things, I guess. I'm just sort of, I don't really know that much about how it works, other than this is what is actually doing the job. And the triggers come in and they get tickled. I don't know what tickles them. And it's just going to have to be a zillion different tickles. The sheath comes off and sheds off. What happens to that? I don't know. I imagine somehow it gets recycled. It's some kind of a material. And just think about the size of this thing. Millions of these little tiny balls inside of one cell. Stunning. And when they unfurl, baby. I'm not talking about a little bit. They get gigantic. Now, you see the blue ones, and then you see the greenish looking ones over here? All right, these green ones, and you see the blue ones. The blue ones are enzymes, and they break molecules apart. They're very, very exotic, and they instantaneously break a ton of molecules. The protein products are the same, come from the same place, a bacteria, but it's one that says, I make things that are for your body to make bones and muscles and eyeballs and earlobes and whatever. And those are the proteins. Those are the building blocks. Those are the building blocks. And then other enzymes actually also put together the building blocks. So enzymes break and build, and proteins are the blocks that the enzymes use. And they're created because the enzymes break up what comes in, and then they use what they broke up to build. It's a dual process situation. Now, let me show you what that actually looks like, how exotic this thing is. And don't forget, we got these balls, millions in a single cell, turning into these huge, huge enzymes. When I say huge, they're bigger than what would be allowed to go through the brain barrier. And the blood-brain barrier says, I will only allow things through this barrier that are smaller than enzymes, because enzymes can destroy things, and they can get into your brain and just wreak havoc. So they will only allow the ribosomes in, and the ribosomes can go in, I guess, quite easily. You can have millions of them in one cell. So you're going to have a freeway to let those go in. However, the enzyme's different story. They're huge, huge, and it says, no, you can't come through. You little guys can come through. When they get inside your brain's going to need them. Your brain needs them just like everything else, but it needs them in the form of a ribosome, floating freely through the system, but once they unfurl, then it's all over. They're gigantic. Hold on a second. Okay, my sterling friends, what do you see? That right there is what's called an enzyme. Now, I showed you what a ribosome does. It is a ball that is like so tiny, millions can be in a cell, and when it unfurls, this is what comes out of those enzymes. Can you imagine? And every single one of these atomic bits has to be stuck together in the correct order. And these are those polypeptides. These are not uncomplicated little packets. These are 20-digit codes. And you got a bazillion of them. And they all have to be exactly in the correct order in order to click to what it has to click to make and break that other thing instantaneously. Because you have literally the capability to probably do hundreds of trillions of electronic interactions that are extremely specific because of the number of amino acid codes that you have. Does that make sense, Fran?
That's insane. I get it. It happens that quickly, and there's that many of them. I mean, they got 10 million of them in a cell, just ready to go unclick things.
Not only that, every one of these enzymes has so many dipole molecular moments in it that the click, when it hits the thing it wants to kill, it's instantaneous. It's a zip, done. And the same thing with breaking down food and so forth. I always thought it would take a long time to digest this and that. I'm starting to think no. I'm starting to think things are digested pretty quickly with this amount of energy, specific energy, specifically targeting specific places in specific edible things. Because life in general, plant life, body tissue, tendon, bone, they're primarily made out of similar arrangements of molecules. So you would need the same or similar types of invasive chemistry to break them down, you know what I'm saying? So I would assume the program in each bacteria would be designed very, very, very specifically to attack a very, very, very specific molecule.
Or designed to eat.
Right, yeah. Break it down to eat it, to destroy it, to disassemble it, to
rebuild, recycle.
Yeah, basically to recycle. Yeah, that's a good way to put it. And they enzymes are found everywhere in the body. I mean, for every single thing, even with breathing, they're used in breathing. They're used in everything. The excretory system, every exchange of everything, and even your blood vessels, your nerves, the electrical signals in your nervous system, all are electrical in nature. They're all dipole electrons working based on some form of a program or an exchange of molecules in most cases. Once life is started and it starts to grow, it becomes an exchange of molecules, basically. And then it works with the program, which is the DNA, but it's got to have the chemicals. And the only way, as it says, the bacteria create the enzymes. You don't have the enzymes, you don't get the chemistry. And these are the things you don't get, something like this. If you don't have that, you can't do a certain job. And they are finding that a lot of the issues that they always considered to be genetic can be triggered by the molecules being methylated or not methylated, which means just turned on or not turned on. And they're starting to understand some of this deep chemistry, but it only happens from enzymes. You die within 30 seconds if you didn't have enzymes in your body, fall over dead. And that's why your enzymes die when your bacteria dies. All right, does that make sense to you? If your bacteria dies, the bacteria that particular strain of bacteria created, that enzyme's not going to be there, right? Would you agree?
You can't make it anymore.
Can't make it anymore. It's not there. You don't have that enzyme. I mean, you don't have the bacteria. So, what does, and I know you know this, but I'm going to pose these as questions to you so that you can respond and we can make it understandable, hopefully. What does a pulse of radiation do? Like a real heavy-duty ionizing, which means it breaks off little electrons. What does
the bacteria?
Right. It destroys membranes. All right. It rips holes in membranes. If you rip a hole in a membrane, you're either going to die or you're going to have to repair that membrane. All right. We as animals can repair our membranes. They're built to be repaired. You can the plant cells, single cell creatures don't have that ability. Once their membranes are done, they're done. They die. So here you are walking around in a nuclear facility, and something goes wrong, and the enriched particles get way too close to each other, and they, boom, a bang comes off them. Of what? What would the bang be? What would you think the bang would be if those particles got way too close to each other and just smacked together too hard? What would you think would be the result of that?
A nuclear explosion?
Basically. But it wouldn't be a nuclear explosion because you wouldn't have enough to...
Well, not... It would still release. It would still release radiation or something. It's
exactly. There you go.
Something in the reaction.
Exactly. What it would do, it would force so many particles, which are already enriched to the point where they just want to get the hell out of town already. And if you smack those together, all the white coating, and not all of it, but a lot of it would just shoot right out of there in ionized, because there'd be particles that would be very they'd burn, and they would ionize and burn through these cell layers and kill them of these and bacteria. It would kill the bacteria. Now, so instantly you get pulsed. Instantly your bacteria dies. How long do you think it takes you to get sick from not having the enzyme? It's only an hour or two. You may not even feel anything when you get pulsed. And an hour or two later you start throwing up, diarrhea, nausea, headaches, all kinds of bloody discharge from your guts, bloating, and it's because you don't have the enzymes. That fast they go away if you don't have the right bacteria. And I got to talk to you about, remind me this, I got to cut off for 1 second here, but remind me to tell you about the enzymes working for autistic kids and the ones that don't take them. Only a day or two, they miss the probiotic that they need to keep these
factory going.
Right, to keep the factories going. They shut down, and then they start getting, right away they go right back to that system. They don't
so their
have...
body is designed to shut down the factories, and you got to keep on pumping the factories up.
Right. Right, but what they're doing is getting probiotics. They're not getting the real living bacteria living in their body, which makes the stuff automatically. And the only way you can get that is to transplant the bacteria. And that's what they won't let you do now. They won't let you transplant the bacteria. I'm not kidding you. Now, hold on 1 second. Hold on 1 second. Okay, this is, I'm sure, has gone very long. Now, we started here talking about these crazy things they're seeing in space during the Tether experiment, and how they got these little cuts in them. They got a dark spot in the center. They're glowy. We know that some of them are in front and some of them are behind the Tether, which is, the Tether is like 80 to 90 miles away from the station that's viewing this. So these are way behind, or at least behind, that 1 mile wide Tether. That's a 1 mile wide. And we determined they're approximately maybe 4 miles wide, something like that. Or maybe wider, I don't know, I have no idea. And I don't know if they're all the same size or not, because some of them are far out, some of them are close. But we've seen this and watched these particles go by. Some of them are coming straight down, some of them are coming sideways. So, like that one there just went behind. Some of them are coming in front, and they have this glowy stuff. Now, anytime you see a glowy stuff like that, I relate that to matter bouncing light. All right. We spoke about this earlier, and we talked about these previously unknown organelles. And these are what they're now calling hemifusomes. And they're different particles that are floating inside the cytosome, the fluids in your body, and this is under cryo electron microscope, which is, first of all, electron microscope is as tiny as you can get, and secondly, cryo is the coldest you can get. So it has stripped off as many electrons as they can. Getting cryo means remove those white particles, remove the ones that are the heat, remove the heat particles, just leave the ones that are not the heat particles. Now, we see these as a certain distance apart. Now, I'm going to zoom in on this. I got Fran listening in, and we've discussed this back and forth, and he may jump in on it, which is, I'm hoping he will. If he's got anything to say, now's the time. All right, here we go. We're going to jump in on this. And don't forget, we've got these different colors, and these different colors are endosome-like vesicles, is the pink ones. Then you got hemifusomes, whatever that means, and then hemifusomes. I don't think they know what they are. They can see these particles. I don't think they know what they are.
And the yellow ones look like the ones up in space.
Yes, they do.
Zoom in, because
yeah,
they got the dipoles on them and everything.
Yeah, it's amazing. But what my point right now is, they're calling them these things, but I don't think they've never seen them before, so they didn't know this. It says this is like discovering a new recycling center inside the cell. Well, what does the recycling center have to do? It has to use enzymes. So now let's look into see what this recycling center looks like. And here it is. All right. Look at these particles. You see these? The little bumps on them? Now these are a certain type of enzyme producing ribosome. I think, in my mind, that's what's going on here. And like they're saying recyclers. To recycle something, you got to break it down. Would you agree?
Oh yeah.
All right, that's what these do. They break things down, apparently, and they are the recyclers. And they are inside the cell membrane. This out here is the membrane of the cell, way out on the edge. You know, if you read through the articles, that's how it talks. Now, so I believe the pink ones are these ENs, the blue ones are MVBs, and the yellow ones are HFs. And in my mind, those are three different types of enzymes, which is three different types of...
Do you think the universe get sick?
Say it again?
Do you think the universe might get sick?
Well, yeah, it's possible. But what would sickness be? Sickness would be an invasion of something, right? When we get sick, we get invaded.
Yep.
Now, how do you react? You got to have something. We get, you know, that would be an interesting thing to really dig deep into, but to say to get sick, I think it recycles itself, and that's what it says in the ancient text. It recycles itself. So that's what happens to these particles. These are the recyclers. So I would say, in a manner, yes, it gets sick, or I suppose it could die. And what do the recyclers do at that point? I guess they just lay around, or I don't know, what would happen if it just died? I mean, the questions go on beyond possibility for anybody to deal with.
We know nothing goes to waste.
It's hard to understand, but it makes sense when you think about it. These are the little recyclers. They've never even known they're here before. They live inside the cytosol, right? The fluids, cytoplasm. All right, and this is what's inside the cell membrane. So it's inside the cell membrane, cytoplasm. And the bacteria are the factories that create the enzymes. And the enzymes come out of the ribosomes, and there can be millions of ribosomes in a single cell, and they work trillions of times faster than they would without that cell. And we had seen up in outer space all of those little things. They look just like those things that we were looking at. These things right here.
Something's floating around up there. Just saying.
Well, we do know the space smells like steak. that's all the astronauts say that. They come in with their helmets and all that, take them off, and they smell like steak.
Zeus likes the smell of burnt steak.
That's right. God said, and they sacrificed and burnt a burnt offering, and he accepted that burnt offering rather than the other offering, as I recall. In the Bible somewhere it says, and even in Zeus, and do you know about the deal with Prometheus?
Oh yeah, he gave him the scraps.
He gave him the scraps, he gave him the bones, and Zeus got pissed off.
Yeah. He tried to trick him. Like, come on, Prometheus.
I guess they did.
Prometheus paid the price for his trickery.
He was the trickster, yeah. Anyway, this brings up some really interesting stuff. I'm going to have to wrap it up at this, but I mean, I appreciate you discussing it with me, and you brought some insight into it, my friend, and I appreciate it.
I'd love to talk to you. We can go all day, man. This is awesome stuff.
Well, we'll do a lot of it because every day something new is coming up, and especially in these other areas of physics and so forth. And like Bennu, you know about Bennu.
Oh yeah.
Yeah, now here's the Bennu. Bennu facts. Potential for life. It's a freaking heart.
It's a heart.
And that's how big it is.
150 miles? What, 140 mile wide?
No, Psyche is 140, and that is also a heart. Bennu is only a third of a mile wide.
Oh.
This is it compared to the Empire State Building. And they landed on there and picked up heart muscle.
They brought back,
Yeah, this is when they landed.
Their old blood. What are they going to make with that?
They brought it back, and it's heart muscle. I've shown this without question. It's heart muscle.
What kind of creature was that from?
One of Earth's also leave us. The answer involves cohesion. Bennu, scientists had expected cohesion to act like a bit of glue between the rocks, making its loose surface more solid. But the tag event showed that Bennu's uppermost layers are nearly cohesionless, deforming under stress like a fluid.
Did you see the colors?
Huh.
Look at this.
But the tag event showed.
You see this? This is when they came down and went through the layers, which is blood. These layers are the layers of blood, and that's iron banding. Now watch this. And now don't forget, it was powdery basically. They were able to blow it away with nitrogen. Watch.
Whose uppermost layers are nearly cohesionless, deforming under stress like a fluid. A good analogy is a ball pit. Although the
A better analogy is blood cells.
plastic balls are solid, they easily slide past one another and past boisterous children, behaving in mass like a fluid.
That's what blood is a fluid. Here we go.
Thanks to OSIRIS-REx, we now know that Bennu's surface is not held together by cohesion, but by gravity, or microgravity, with a minute tug less than 100,000th the pull of Earth. On the Moon, gravity is 16% as strong as it is on Earth, and more than 16,000 times stronger than it is on Bennu. As a result, loose material in the lunar subsurface is packed together more tightly, making the Moon's surface relatively firm. If a 50-kilogram mass of solid iron were to hit the Moon at the same speed as the tag event, it would sink into the ground by only half a centimeter. Repeating this experiment at Bennu would yield a dramatically different result. Though the mass would strike with the same force, it would plunge 17 centimeters before stopping, over 30 times deeper than at the Moon.
That's because that Bennu is coated with blood. That blood has leaked out of all of the vascular network, which is extensive in the heart. And the blood has leaked out. And it's still going to be wet. They found clay inside there and sarcomere.
Moon. Bennu has consistently defied scientists' expectations, as each new finding reveals another facet of this small but surprising world. Using data from OSIRIS-REx, we now have the ability to look back and accurately recreate 30 seconds on asteroid Bennu. On October 20th, 2020, OSIRIS-REx made its final descent to a sample site called Nightingale. With its TAG-SAM arm outstretched, it approached the surface at 10 centimeters per second, the walking pace of an insect. 1 second after contact, it released a canister of pressurized nitrogen, detonating an explosion of particles and driving material into the TAG-SAM head for sample collection. 6 seconds after contact, while it was still sinking into Bennu, OSIRIS-REx fired its thrusters to begin the back-away maneuver. The engine burn lasted for 24 seconds, continuously pushing against the spacecraft and rapidly slowing its descent. Flying debris from the thrusters and the gas release pelted the science instruments, clogging them with dust. 9 seconds after contact, when OSIRIS-REx had sunk nearly half a meter into Bennu, it reversed course and began to rise. At 16 seconds, the TAG-SAM head re-emerged from the subsurface as the spacecraft continued to accelerate.
I just want to say one thing. I'm sure this is AI showing this. I'm not positive, but it's representative of exactly what I would have expected. And I saw the colorized version of this, and it was the true version. And it was the red, the black, and the yellow of blood, iron oxides. Now I'm just gonna, you got any questions yet, Fran?
And they brought this back?
Oh yeah.
That flew back.
Yeah. I'm gonna show it to you. And it's heart muscle. Watch this now.
30 seconds after contact, OSIRIS-REx shut off its thrusters and drifted away with its sample of Bennu. Almost 6 months later, on April 7th, 2021, the spacecraft returned for one last flyover to observe its footprint. At the point of impact was a new crater.
There it is right there. This is the red, the black, and the yellow. You see this? That's not fake. That's the black is the hardened up blood. We know that happens, right? Do you agree with me?
Yeah.
So these are the hardened black parts. You see all around here, they're showing this red. That's the blood cells. And the
Where's the iron at? Which one's the iron?
Red is the hematite, which is the red blood, and the black is the magnetite, which is the black blood. They're both iron. They're two different states of iron. And when you do, how you do with iron, and as with all metal ores, is you heat them up and you burn off all the stuff to a point where you have just iron left. So it's just smelting. And you have to get hotter and hotter and hotter to get more and more refined steel and carbon until you get up to where you basically got carbon left. Carbon steel is that's the tough stuff. But right here, they're going down to the bloody surface of Bennu. This is just nothing more than a bloody surface. If you look over here to the right, and you see this black and yellow and red, that's exactly what the colors of blood are. And this is exactly what happens. The black gets hard, the red stays sort of fluidish and dusty.
It looks like a scab.
Yeah, I have a iron meteorite here. No difference whatsoever. Identical, same colors, everything. The black, the yellow, and the red, identical. And it's got the big holes in it. Mine's a lung. And it's got the latch on the end of the lung. It's got all the blood vessels. And it actually, I think it still may have some catalase in there. I think I did get a catalase reaction one time. Anyway, this is a heart. Now, they said they picked up some stuff and came back with it. Well, let's see. Okay, I am claiming that Bennu is a heart, and so is Psyche. Now, this is the National Science Foundation, along with the Smithsonian and a bunch of other of our money spent to go out to Bennu to retrieve this sample. And they're touting how they're going to be studying this for many, many years, and how can we better understand the diversity and characteristics of Bennu by examining these features of various spatial scales and all this business. And this is all by these top people, and they have the samples. Now, let me show you the samples they brought back in relation to biology. Okay, once again, we're looking at things that are in space. This just happens to be Psyche, which is another heart that's in space. This is much bigger than Bennu. Bennu is only 1/3 of a mile wide. This is 140 miles wide. That's Bennu right there. It has tubes coming out of it. These are not craters, they're tubes. And these are not craters, they are valves. And this is not just a ridge line here for no reason. That ridge is there because it's a tendon enthesis which holds the muscle. And these colors are here because these are the colors of oxidation states of blood. Red, yellow, black. As we saw on Bennu. Now, that's a heart. This is the tendinous stuff that's still there. Above that, right here, is where all that plumbing just falls right off. And it falls off because of iron banding, which there's a red and a blue line straight across there, which is the two. See over here? Right above the valves. There's red and blue blood, which they don't mix. They don't like each other. They push each other apart, and the tubing just pops right off the top. And you end up with what you see right here at the bottom. And this is what we see in these veins, in these valves. And you see muscle, and then you also see some tendon. But you see right over here? I don't know if you can see that clearly, but that line right there,
I see it,
is where they break right off. And the autopsy guys tell me the same thing. They say, yeah, they just come apart when they've been, the bodies, they do science. So the bodies lay in storage sometimes for a while, and they just come apart because the blood now doesn't move. When it doesn't move, it pushes. When they're moving, they're just moving. One goes past the other. I don't know if they have any effect on each other, but they're moving, so it doesn't create layers. All right, so I don't know if all that made sense, but this is Psyche, and it's nothing but metals. That's why they're so interested, because they think they can mine these things. So we see what we're looking at now. Let me show you something. All right, I showed you Psyche, which was the heart in space, 140 miles wide. Now, Bennu is not nearly that wide. It's 1/3 of a mile wide. However, it is clearly a heart. Now, what I can show you about this is the valves. Now, this valve right here, if you follow my cursor around in a circle, follow it around. Now that is a valve. Now what does that valve do? it lets blood come out towards us. You see that little arm right there? You see that arm right there? You see that arm right there? This is, I believe, what they call a tricuspid valve. And it lets blood come out this way towards us. But if we try to force blood back into it, the valve clamps off with these three little arms coming down, closing that hole in the center. You understand, Fran?
Yeah, vein and artery. And
It's
it only goes one way.
It's a one-way valve, yes. Now, this also is a one-way valve, but it's the other direction. Now, I just noticed something that I didn't notice before. It's these two holes. I think it has the same two holes up on Psyche. I remember seeing something in there, and then there's this big chunk here coming out of what that right there. You see this? This is another huge, I believe that's the aorta, and it's got a chunk coming out of here, which was probably the attachment, one part of the attachments that held this to the plumbing up above. You know, they break in all the different ways. They might have broken flat as a pancake and other ones, maybe not. But that's right on the edge of this biggest one. Now let's go back. Say it again?
I said they just got the picture upside down.
Basically, yeah. It's, you can look at it from any way you want. I believe the blood primarily is down here. That's where the bag of blood sits down in all of this sac here. And above this would have been where the plumbing just glued on there, right, glued right to the top, basically. Now, yes, but you can see what I'm talking about. Now, let's just go back to Psyche for a minute, because these two holes here, I thought I saw those in the other one. Now, hold on a second. All right. This big crater has that stalk coming out of there. Now, whether that's something similar to what we see on Bennu, I don't know. Again, this is Psyche. Now, this other valve has two spots on it. This one here and this one here. And these other two here. And this has two, one right here and one right here. Now, this is too much of a coincidence to be a coincidence. Now, this is the tube coming out of here. Then there's another tube down at the bottom, right here. And this abrupt transition is between an artery tube and a vein tube, and they push each other apart. They don't like each other. The black pushes the red away. And the red becomes powdery and stays fluid and rehydrates almost instantly. I've shown this stuff so many times, it's amazing. Now, so they're both hearts. As far as I'm concerned, you can't prove to me that's not a heart. And if you call that a crater, then I got problems. Why does nobody ever mention any of this stuff? And why does it get laughed at when it's brought up? Like, oh, that's pareidolia, don't talk about that stuff. You're a crazy guy. No, I'm not. I'm looking for truth and reality. And that's what I'm trying to find. And so is Fran. And we're looking down inside here. Is this biology? Yes. That's biology, my friends.
Rocks are body parts.
So gigantic and in space. And the only thing I could think of is they are either from battles in the heavens, or they are from where Mars was impacted by Venus.
And they just got scooped up and flown out.
They were just splattered all over, because Earth is covered with creatures that have these size body parts. This is not an unusual situation to have this size of a body part. We do know about Typhon in the desert. He's 1,100 miles long. he's easily got a 100-mile heart. And we know about Quetzalcoatl, we know about Scorpio. All these people think we're crazy, but no, we're not. These things are real. They're hard to deal with, but to deal with them by just making fun of people that present them is not the way to deal with science, or reality, or history, or any of that stuff. Whether it means anything, I don't know. Whether it means anything to you, I don't know. Whether it means anything to me, well, it does. Whether it means anything to you, I don't care.
That could be a dragon. That could be a dragon heart in space. That means something to me.
It could be easily. Well, we know that the Earth is just covered with them. I mean, absolutely covered. And they look at these things and they just say, "Oh, those are called flat irons." Well, you've seen the flat irons. They're nothing but dragon scales. They just can't accept the truth that stares them in the face.
And pyramids.
Very, and they are the teachers, though. This is what bothers me. I don't care if the average guy sees this or sees that, couldn't care less. But ones they're teaching, especially they're charging the kids what they have to say. And then they have to say what they tell them to say or they fail them. So that's not a good system. Especially if you're one of the students. The other way it works perfect.
You're paying for lies.
Well, you're paying to be indoctrinated. And if you don't say what they tell you to say, they will fail you, first of all, and not give you your paper that says you're smart. Because you couldn't.
Get a good job.
Yeah, you could, and then you won't be able to get a good job because they say that the kid just can't repeat. And he's no good. And or she, it doesn't matter.
It's art in space. We got to fail them.
Yeah, well, this I told you about what happened to me with my professor. First, when I came out with the Dipole Electron Flood theory, I didn't call it that back then, but I said that everything is made out of dipoles. This is nothing but dipoles, and it's atomic vapor. I said, how do you think this stuff gets from the sun to the earth? Well, it's perturbation. I said, come on. I said, there's particles. And it's so obvious, it's unbelievable. But if you have to work with Einstein's theory that light never slows down, which is absolutely ridiculous, and they know it's wrong now.
They paid them for it. That's why we've got to work with it.
Well, if you don't say that, then they destroy your life. Your friends will turn against you instantaneously. If you go off, you go off script. You're done. You have to say what everybody else says, and then some guy sooner or later, like me, starts shaking a boat here. Sooner or later, they're going to have to address this. And I mean, I'm talking. Velikovsky died, and he was considered, and he still is, a pseudoscientist wacko. I wrote my book about him in 2015 because of his treatment. And he was the one that talked about worlds in collision, which a lot of my research supports. And he's still considered a pseudoscientist, which means nothing he said was right. He's just a guy making things up. Now, the funny thing was, I put in AI, am I considered a pseudoscientist? And they say, no, I'm just considered a self-published research or something like that. It wasn't as demeaning as it was to Velikovsky. But Carl Sagan was the one that attacked Velikovsky. Really, he was a very, very, very arrogant guy. And they crushed him until he died. So
that's what you do when you got the truth.
They forced his book to be taken off the bookshelves after it was on #1 for 11 weeks in a row on the New York Times bestseller list. 11 weeks in a row, #1. And the academics told the publisher if they didn't take it off the bookshelves, they would never buy another book from that publisher, who had it was Macmillan, and they had a big publishing to academics. So they caved, and they said, okay, we'll take it off the bookshelves. And they did. And
Gatekeepers.
yeah, and they ended up Doubleday, which was another publisher, but they didn't have any academics to publish to because they considered them cartoons or something probably. So they took it on, but by that time they had pretty much turned him into a buffoon, and anybody that would read that garbage is insane, and and all the teachers would drill that into the kids, and they had a core of kids that would not accept the crap that the teachers were feeding them, and of course they got destroyed basically.
Oh, they put them in insane asylums.
Yeah, well, I've had people and I've had people tell me that I should be evaluated for this and that, and I've had other people in other countries. They actually, and I don't know if they actually did lock them up, but they threatened them with, they had to take medication, that type of thing. And I've had a lot of people tell me that their wives or husbands have told them that, they got really serious mental issues, that they got to be. They should really have somebody talk to them, and all kinds of very, very.
Foreseeing with their eyes.
They just can't see. They can't see. They don't want to see, and they can't see.
Well, it's what you said. This is for you to see and others to test.
It is a test. I never thought it would be. I thought if you had enough evidence that was undeniable, it would not be denied, and that is just not the case. Has nothing to do.
Scans and DNA tests done, and you came back with proof, and they still say, nope, that's not the proof.
Oh, I didn't show you the proof. Where do you see the proof? Hold on. It's muscle sarcomeres. We know it's a heart, and heart is made of almost entirely of muscle. Now, where do you see this? Okay. Now, I don't know if Fran's seen this yet. These are the samples that they picked up from Bennu. We just saw how they went way deep down inside, and they just smacked something, and they pulled up this chunk of stuff, and this was some of it. All right, it's small. This is 1 millimeter size. This bar. Okay, so this is small. Because heart muscle and sarcomeres are small, and that's what this is muscle sarcomeres. Now, this white stuff over here on the top left panel is, in a certain light, it shows that this phosphors here, phosphates, and they glow, and there's layers of them. Now, if we move over to the right, it's the same as the thing on the left, but we're not highlighting it, but we can still see those little white flakes, which are the phosphors. Now, then it goes down to the smaller block, which is here down at the bottom left here. Now, we can see the phosphate layer, the flat plate at the bottom with the little white flakes on it, and then this blocky looking thing on top of that, and then on top of that blocky looking thing is this layer, which is another layer of the white stuff, and then on top of that is another layer of the blocky stuff. So we've got layers here. We've got a layer of phosphates, a layer of what's clay. This is clay sarcomeres. Above that we have another layer of the phosphates, and above that we have another layer of the sarcomeres. Now, what do the sarcomeres look like, and what the hell is a sarcomere? Well, that's what the sarcomeres look like. Now, sarcomeres are these little blocky, boxy looking things, and what they do is they contract and pull amongst themselves where there's connective tissue, and that is what makes your muscle function. They contract, and that's how a muscle, and then they relax, contract, relax. Now, this up here, if you can see here, this is the muscle bundle. Cardiac muscle is bundled and all floppy looking things that go in and out of each other. They're bundled cords, and this is it. This is what it is, and it wraps around your heart, and they block into these blocks. So this is like a cross cut, and this is like what they look like as they go down and make the sarcomeres. Now, let me show you what a heart muscle on Earth looks like. All right, this is a heart attack victim. These are the sarcomeres. these little blocks. Now, this stripe across here, this right across here, this right across here, separates those blocks of sarcomeres. Now, what we saw was this, these little red layers, which are almost like, well, they are clay. They literally end up being clay. Now, these areas here appear to have vaporized in the ones that came from Bennu and left the red blocky ones. Sometimes it goes the other way. Sometimes the red blocky ones go and all you have is a connective tissue. I've seen it go both ways. But we do see this layer of phosphates, and this is phospholipid layers. Lipids means fats. Phospho means phosphorus. Phosphates are phospholipid fatty layers of membranes. And there was one here, then a sarcomeres, and then another one, and then a sarcomeres, and then another one, a sarcomeres. And that is exactly what we're seeing here in a human heart that's here on Earth. Do you have anything about that you want to say, Fran?
That heart just got ripped in half.
Yeah, these are the places that let go. This place let go, and here, down here, and over here.
Looks like it caved in.
Well, it ripped.
Right, but they're all separated. What I'm asking is, it's not one big piece. They're like in strips, so they're able to slide and rip and then rip.
They slide each other among these layers because it has to twist and to distort and go to the right and to the left and up and down. Your muscles go every different direction. So the plates of muscles they come together. You see where my cursor is over here?
Yep.
All right, that is one side of the muscle sarcomere. This is the other side. When they engage, this piece right here slides into this piece right here. That would be a constricted muscle. It would be like about a little more than half its size. And in between every one of these little red things, there's a million little connective tissue lines. All right, they're the connective tissues, and they're the things that do the real pulling. But the red stuff is it's the reactive stuff. The others, somehow it pulls. It pulls this plate into that plate. It slides it right in, and that's what a constricted muscle is. But what we saw in the sarcomeres from Bennu is just the red stuff. And in between, it looked like this stuff had eroded out for some reason or cooked out, whatever. Because it's different. It's a different material. But we did see these blocky little blocks that are now clay. And we did see the white stripes, which are the phosphate layers, which are phosphate layers here too. So we see these, and we see the phosphate layers. But something here bled out, or leaked out, or cooked out on the edges, and left the blocks in the middle. Let's go back to that one more time so we can see it again. Now we've freshened ourselves up to understand what we're seeing. Okay, Fran and I are going to be doing a lot of meetings that are going to be speculation, thinking about this or that. This I wanted to present as the material evidence, and the agreement we had was if he had any doubts about what I was saying, or any concerns or any questions, to just interrupt. And you see there was very little interruption. And he said, I don't know what to say, because there's nothing I can say to go against what you're saying. Which is, there is really not much you can go against this. Now, what we're going to be next doing is more of an interactive between Fran and me, and some other people too. But Fran and I could be doing a lot of this. We'll see how it goes. and it's going to be an interactive thing this time because a lot of it is speculation, and he's got a lot of knowledge on the ancient texts. And he's followed me for a very long time and has watched virtually all my videos, and he's got a good memory. He reminded me of things that I said I forgot I said. So he's going to be very helpful. Anyway, I want to thank Fran, and I want to thank you all for watching. Stay tuned. It's going to get real interesting real quick.